organismal things that influence microbial growth, or how other organisms can influence the growth of microbes. In a few situations, we're going to have microbes live in shared habitats. Almost every single microbe is going to share the environment with another microbe or another organisms. These associations are going to be between similar microbes or similar species and dissimilar types or dissimilar species of microbes. Sometimes it's a multicellular organism, such as an animal or plant, i.e., think of a pathogen making a human sick. That's the one you probably care the most about, since we have a lot of aspiring nurses in this class. Or we could have sometimes an interaction between a microbe and a plant. For those of you who have an agri-science background, hey, maybe that'll float your boat. These interactions can sometimes be good, sometimes they're bad, sometimes we don't care at all. They have no particular effect. Sometimes these interactions are obligatory. They are necessary for one of the organisms in this relationship to stay alive. Sometimes it doesn't really matter. Sometimes one of the members could care less about this interaction. Oftentimes, these interactions are going to have some kind of a nutrition exchange or nutritional exchange where macronutrients will travel from one organism to the other or back and forth between the two. Here's a classic example of two macroorganisms interacting with each other. Here we have a gazelle, an antelope. I'm not actually very sure what my large mammals in Africa are called. This is an African mammal, and it has some birds that are picking insect parasites out of its skin. It's a mutualistic relationship that's good for both organisms. The birds get a dinner, they get a snack by eating the bugs, and then you can see a bunch of bugs down here in the corner, and then this large African mammal doesn't have as many bugs parasitizing its body. So it's a win-win. We aren't going to spend a lot of time talking about macroscopic interactions. We're going to focus more on microscopic because this is a class on microbiology. If we have a symbiosis is just a general term. It's just two organisms working together in a close relationship. A symbiont is a generic term that refers to a member of a symbiotic relationship. And the symbiosis is an umbrella term that refers to organisms having a relationship. It has no specificity as to whether or not this relationship is good, bad, or neutral for the organism. So let's talk about the three main kinds of symbioses that we can have. We can have a mutualism. These are microorganisms that help each other out. This is a you scratch my back, I scratch your back kind of relationship. For example, this is a mutualistic symbiotic relationship. Or we can have a commensalistic symbiotic relationship. In a commensalistic relationship, one partner benefits and the other partner is neither harmed nor benefited. It doesn't really care. A good example of a commensalistic relationship would be the mites that live on our eyelashes and eyebrows. Those little mites, microscopic mites that live in all of our eyelashes and eyebrows, love eating our dead skin, and they just gobble it up. It's a wonderful benefit for them. You and I don't really care that much about them. And it's not like they hurt us, and it's not like they help us either. It's a commensalistic relationship. Another good example would be the trillions upon trillions of E. coli cells that live in our large intestine, or our colons. Most of those E. coli neither harm nor help us. Just kind of there, so to speak. We can also have parasitic relationships. A parasitic relationship is a special kind of symbiotic relationship where one symbiont benefits and the other symbiont is harmed. Generally speaking, parasitic relationships are what cause diseases when a microorganism causes disease in a human being. Sometimes we can have an antagonisms take place, and this is when a free living species arises when members of a community compete against each other. So it's not necessarily that these two microorganisms are directly interacting with each other, but they're competing for the same resource. And here's a good example. Let's say there's two people in the room and there's one donut on the table in between them. They both really want to eat the donut. But if person A gets to the donut first and eats it before the other person can, the other person's going to go hungry and feel bad and sad because they didn't get to eat their donut. That's an example of competing for a common resource. because donuts are a vital resource in our communities. Now, in antibiosis, it's going to be when these microorganisms produce inhibitory compounds, i.e. antibiotics, or e.g. antibiotics, and these compounds help to destroy or kill off the competition, so to speak. These microbes that are first to colonize the environment have a huge competitive advantage because they rapidly are going to occupy the spaces that are available and start utilizing the nutrients that are available before there can be other microorganisms that can initiate an antagonistic relationship. If we look in the soil, where we have lots of mixed communities competing for space and food, you're going to find all kinds of chemical compounds that form antibioses. They're fall of 2015, I just read a new article about a fresh, brand new kind of antibiotic that was isolated from a soil bacterium. And right now, if you were able to isolate our soil bacterium and grow them in culture, which is a whole separate problem, we'd probably find many different kinds of antibiotics that have yet to be known to medical science because there are so many mixed communities having antagonistic relationships with each other in the dirt, in the soil, so to speak. Sometimes we can have a synergistic relationship. These two organisms do not need to live together, but when they're present in the same environment, there's slight benefit to each other. They kind of help each other out. Generally speaking, when we have a synergistic relationship, the net benefit to these two organisms is greater than if each organism was living separately or individually by itself. Common synergistic communities include the plaques or biofilms that form in our mouth that lead to gum disease or cavities, aka dental caries, and some of our bloodstream infections also are going to involve mixed communities of bacteria that interact with each other synergistically so that there's greater net effect as opposed when they work together, as opposed to when these bacterial species work separately from each other. So if we looked at a biofilm, that's going to be the example, above all examples of a synergistic relationship, the epitome of a synergistic relationship. It's a mixed community of bacteria and all other microorganisms that attach to a surface or substrate and then attach to each other. And these microorganisms will have pseudo-specialization within this biofilm community. The initial microorganism, or the pioneer, will initially attach to the surface, and then it'll start to secrete a glycocalyx that will recruit other microbes, and those other microbes are then going to secrete additional substances that attract additional microorganisms, and we then have a complex community of microbes interacting with each other. In some ways, you can think of a biofilm as a primitive tissue. It's still formed by microorganisms that can live separately from each other, but when they're working with each other, they change their form and they change their function and consequently increase their rate of survival. Within the biofilm, there's a phenomenon known as quorum sensing. And for those of you that have any kind of parliamentary procedure in your background or have any kind of political background, or you know from Robert's Rules of Order that in order to maintain a quorum and proceed with your meeting, you need to have typically 60% or 2/3 of all voting members present in the body to proceed. That's a quorum officially. A quorum is when you reach the threshold minimum for initiating activity. With quorum sensing, when we reach a threshold minimum concentration of bacteria, all of a sudden, when we go above that concentration of bacteria, the bacteria start acting differently when compared to when they were below the concentration or the population concentration. For example, many species of E. coli at low concentrations are not pathogenic. But if you increase, or I should say strains of E. coli, not species of E. coli, if you increase the concentration of E. coli above a certain threshold concentration, all of a sudden that population of E. coli will switch from being non-pathogenic to being pathogenic. They start to act differently when they reach the quorum. In a biofilm, we have lots of quorum sensing because we have very high concentrations of bacteria. Many different bacteria And strains of bacteria are squeezed into a small area, and different parts of the biofilm are going to have different chemical environments. Generally speaking, there's a higher oxygen concentration on the surface, and then a very low concentration of oxygen in the center or middle of the biofilm. These biofilms, generally speaking, have very thick slime capsules on them, which make it very difficult to eliminate a biofilm with antibiotic treatment because the antibiotics cannot penetrate the slime coating of the biofilm to get to the bacteria in the center. If the bacteria in our biofilms are exposed to these different chemical environments and are reach that minimum threshold for quorum sensing, they start to behave very differently than the free-floating or planktonic bacteria. And the reason they behave differently is because when they reach that threshold for quorum sensing, they're going to activate different genes, deactivate other genes, and are going to exhibit very different structural and physiological characteristics. So here we can see a four-step plan of biofilm formation. We have first a pioneer bacterium colonizing the surface, and then they secrete a glycocalyx that recruits additional bacteria, and then tertiary and quaternary bacteria are recruited to the biofilm. We reach to quorum, and now quorum sensing has taken place. So now specific genes are now activated in the bacteria, and they start to behave differently. And now we have, you can think of it as kind of a barracks or a home base, and this home base, or biofilm, is constantly excreting high numbers of bacteria and regenerating them within a slime capsule that's resistant to antibiotics penetrating the slime capsule. If biofilms form in human beings, these biofilms, generally speaking, are very difficult to treat and cure. So, concept check class. Which of the following describes an association between microbes in which one is benefited and one is harmed in some way? So one benefits and one is hurted. Would it be a mutualistic, would it be a synergistic, would it be a commensalistic, a parasitic, or antagonistic relationship? If you don't know, that's okay. Rewind the video, flip back in your PowerPoint, find me the answer. 1, 2, 3, 4, 5. The correct answer is D, parasitism, or parasitic relationship. If you have any questions, you can feel free to shoot me an email or post them on the discussion board. Happy studies!